Good afternoon, everyone. We are about to start. My name is Maria Schatz, and I'm a scientific program manager for the NIHS Office of Data Science. It is my pleasure to welcome you to the ELK seminar series. By the way of background, ELK stands for Environmental Health Language Collaborative, and our mission is to advance integrative environmental health research by promoting access, use, and harmonization of data through interoperable terminologies and best practices. You can become involved with ELK by and learn more at the ELK website. You can check our ELK resource catalog at this website again. You can join the ELK email listserv using this link indicated on the slide, and you can also contact ELK directly, either through emailing myself or Charles Smith, who is a director of the Office of Data Science, or through this short email at elk@icf.com. And with that, I'm happy to introduce our speakers today. Darius Boost is a data scientist at the NIHS Office of Data Science. He is a bioinformatician with a deep commitment to advancing genomic and environmental health research. He earned his bachelor degree in applied mathematics from the North Carolina A&T State University, and then he went to complete his PhD in bioinformatics and computational biology at UNC Chapel Hill. During his graduate and postdoctoral training, Dr. Boost worked extensively at the Renaissance Computing Institute, RENCI, under the mentorship of Dr. Kirk Wilhelmsen and Dr. Chris Bison. His research spanned several domains. His thesis centered on multimodal meta-analysis of predicted gene expression in addiction and Alzheimer cohorts. His postdoc consisted of building a knowledge graph of synthetic population data and historic flu data to predict high-risk areas in the event of a hurricane. His areas of expertise include genomics, data analytics, and meta-analysis, and his current work involves leveraging HCAP, healthcare cost and utilization project data, to transform raw data into more efficient database structures, with a particular focus on how patient health data interacts with environmental exposure data. So having said all that, Darius, you are welcome to share your screen, and the podium is yours.
Hey everyone, thank you for that introduction. Good afternoon, and thank you for the opportunity to speak today. My name, like was stated before, is Darius Boost, and I'm excited to share with you how knowledge graphs, or KGs, how I will sometimes refer to them during the talk, are transforming the way we integrate, explore, and act on biomedical data. Today's talk is designed to be accessible for whether or not you have computational background or not. And we'll begin with the foundational concepts, look at some real-world tools and examples, and then move into some technical side of making knowledge graphs interpretable and reusable across the biomedical domains. All right. So we live in an era where data is not the problem, it's the fragmentation of that data. Across biomedical research, we have a rich but siloed data sets. EHRs, genomic databases, drug repositories, literature, environmental data, and more. Imagine trying to connect patient health records, genomic biomarkers, and exposure data across 3 studies. It's technically possible, but practically painful with my experience. The fragmentation cost us time, reproducibility, and insight. Each of these types of different data sets were designed with specific purpose, often in its own format, name conventions, and access methods. The challenge isn't just having the data, it's connecting the dots across it. The fragmentation makes it hard to ask integrated questions like which environmental exposures are linked to disease that share common genetic bases. That's where knowledge graphs come in. They bridge silos. A knowledge graph, what is a knowledge graph? A knowledge graph links entities or nodes, like so, through meaningful relationships that are edges, like so. It's like a mind map, only machine readable, structured, and computable. So, what exactly is it? A knowledge graph is a structured representation of facts where entities like diseases, genes, drugs, or phenotypes are nodes, and their relationships between them, like treats, associated with or expressed in, are edges. It's not just data, it's data with meaning, context, and connections. Unlike a flat table, which lists data, it's data with meaning. Oh, unlike a table that which lists data in rows and columns, a knowledge graph captures complex interrelationships. This format makes it possible to traverse across domains from symptom to a disease to a gene to a chemical compound, all backed by evidence. To think of a knowledge graph is to think of a conceptual map where nodes and entities like diseases, genes, and edges and the relationships between them turn static data into living structure. When we think about data storage, traditional databases come to mind, tables with rows and columns. These tables are great for structural data like patient records, where each entry is discrete and consistent. But when we want to represent complex relationships between data points, like how a gene interacts with a drug or how environmental factors affect disease risk, tables kind of fall short. Knowledge graphs allow us to represent this rich, interconnected information with flexibility. In a table, if you want to know how a drug affects disease, you may have to search through multiple tables, make joins, and still miss context. But with knowledge graphs, you can directly traverse from node to node, from disease to gene to drug, without losing memory or meaning. This flexibility and interconnectedness is key for biomedical research, where every discovery opens up new connections. So what can a knowledge graph do for you? You don't need to be a developer to benefit. Knowledge graphs can help scientists, clinicians, and analysts explore data in a flexible, structured way. Now that we've seen what graphs are and how they differ from traditional tables, let's talk about what they can do. First, they integrate data across domains: clinical, genetic, environmental, chemical data sets. You can explore questions across these boundaries. Second, they are queryable in powerful ways. You can ask what drug targets, genes associated with diseases are that share a phenotype with asthma. And I'll repeatedly go to asthma as a example for demos to come. Third, uh, knowledge graphs are expandable and fair. They allow continuous addition of new data and support findability, accessibility, interpretability, and reusability. Most importantly, they enable hypothesis generation. With a knowledge graph, you're not just looking at facts, you're discovering new connections. Let's look at a few examples where this can be really exciting. So this is a real world example, how genetic, looking at asthma, and let's ask the question, what factors contribute to asthma and what interventions are most promising? This is some kind of results you can get back from using a knowledge graph. The question spans several domains, like I said before: clinical, genetic, drug response, environmental triggers. And we can make the connection now with this knowledge graph. We can trace relationships between these domains. We can connect air pollution exposure with asthma exacerbations to genes involved in the immune response to drugs that modulate those pathways. Let's say we are studying asthma, and it shows that elevated PM2.5 genomic data suggests that the IL-4R involvement. How do you connect these threads? This is through knowledge graphs. Makes it possible. All right. So here I'll go through a demo. The demos of for the first knowledge graph, biomedical knowledge graph adjacent tool called the Monarch Initiative. And the Monarch Initiative is a powerful resource that links genes, diseases, and phenotypes and model organisms. So I will click here and hopefully it'll continue to share my screen. Let's stop sharing here and reshare. Okay. So we'll do a quick demo. And here we'll look up a disease. Let's say asthma. What we're given in return is we'll see the description of the disease, and then we'll get a few other things that are of interest. We'll get links to related genes, to disease phenotypes. We can go down here with our first results. It lists the different phenotypes associated with asthma, what they where they're categorized, so their respiratory, immune, or nervous system. The disease to the phenotype. It lists other diseases that are similar to asthma that has a phenotype that are related to it as well. Here we have disease models, so it has different studies that are done in different model organisms. So we have, let's click on one here. This is in the house mice, and it shows that it has this phenotype and the phenotypic feature that is associated with asthma that is studied in this model organism. If you wanted to go further, you could go into your details. Into details, it also lists the publications where this information can be found, what this is provided by, which is a little bit of backend for the knowledge graph itself. And this link will point you to another page within the knowledge graph to give you more information. Genotype to gene. So it talks about the gene and the genotypes that is found in the mouse or model organism here. Then we'll go back to the original page here. And yeah, so then it has a hierarchy of like the superclasses and subclasses for cough variants and intrinsic asthma, and then those varieties like so. Now I will go back to my presentation. I'll be making a couple jumps here in a second. All right, so here would be a video demo that kind of talks about this researcher's experience. This demo will be, or this video will be live with the presentation in post-production, but it's a short video clip, talks about his experience, his personal experience really, with using the Monarch Initiative and how it helped him diagnose his son who was suffering from an illness. All right. So for our next demo, we're going to look at the NCATS Biomedical Translator. And the Translator project, in the Translator project, you can ask the biomedical question in natural language, and it maps to a graph showing connections and evidence. The NCATS Biomedical Translator is a tool that allows you to ask complex biomedical questions, and it builds reasoning graph across diverse data sets to answer them. It's part of the NIH's effort to make biomedical knowledge computable and interpretable across domains like genes, drugs, diseases, phenotypes, and exposures. All right, so I will go into another demo here with this one. Stop sharing. So with NCATS or an NCATS translator, you can see it has kind of a structured system already where it will provide you some questions you can ask. So we can say what drugs may treat conditions related to, and then enter a disease or a phenotype. So for us, let's try Alzheimer's. And as you see, it loads, and they can kind of give you a predicted answer here. So let's go Alzheimer's and go here. And then down here, you can kind of see the workflow. Select the relationship to explore, review the select results, and analyze the evidence in the workspace. Let's click go ahead. Usually shouldn't take too long. Had this up before and I did it. It took a couple, maybe a minute, but you shouldn't take too long. Let's see. There we go. So here we can see the results, about 243 results, and we're looking at drugs that may treat conditions related to Alzheimer's. It has your name of your drug here, and then other information that it's providing. So it has relationship, this treats this, and it's in clinical trials here. Also, if you click on the tab treats, you can learn more about the reasoning behind this, where AK got this information from. So let's see what this says. All right, so let's go here. Should give you some background sources. Here we go. So you can see the clinical trial that is being used, that is the rationale to provide that information for Alzheimer's and the drug being used here for Alzheimer's. Now, this can be like a starting point for some people. Again, you can say, okay, what you're looking for this. And then if you wanted to go back, you can say, let's go back to their homepage. Instead of it saying what drugs may treat, you can look at what genes may be increased by a chemical. And then if you have the chemical name of that drug, put that drug in it, then you can see there may be some overlap between those two things. So for each related disease, explore the drugs that are used for treatment. This can uncover potential repurposing for opportunities for things like Alzheimer's or for asthma, for example. The key takeaway for an NCAT translator is that it integrates data across various biomedical domains, facilitates the discovery of non-obvious connections between diseases and treatments, and the platform supports hypothesis generation. Sure. Back to my presentation. Actually, instead of going back to the presentation, I'll just go to the next slide in the presentation, which would be sharing my other demo, which is going to be RoboCop. So give me 1 second, I'll share that. Okay. So now we're looking at RoboCop, which is the reasoning over biomedical objects linked to knowledge-oriented pathways. It is a tool that lets you ask biomedical questions like the other ones in a natural language, and it returns structured answers powered by a knowledge graph. It uses data from trusted biomedical sources and the translator infrastructure that I mentioned earlier. You don't need to know anything about ontologies or graph theory to use it effectively. So just kind of walk through a simple demo. Let's ask it a question here. So we'll say, okay, we want to look for a gene okay. And they want to see that it is associated with, let's get rid of related to, and associated with, and then let's pick okay. So we'll submit this, and this shows us one result here. But if you expand this, it shows these three different links. It says that this is associated with asthma or genetically, and that this occurs here. So if we click on any of these edges, let's click on occurs with in literature with, it shows you that it has about 81 literature um, references that counts for how many times it shows up together between this gene and asthma. You can look at genetically associated with, and then, oh, that's bad. Let's scroll up a little bit. So here it can show you some sources of where the knowledge comes from and how it's built into the system. This isn't necessarily useful for anyone. Let's see if it has the links in here. Let's try a different query. Let's go with associate this with disease. All right, so what we have here, we're just using this gene and associating it with a disease. And then you can see that it has different diseases that are listed here. Let's try neoplasm. And you see again that it's positively correlated with it or occurs together in literature. If we click here, again, it shows that it shows up in literature 1,118 times and positively correlated. Aha, here we go. It shows you some BioLink publications that you can click on that will further support this as being true or not. So this is just ways that you can go through, and you see there's not just neoplasm, but many other diseases that are being linked or associated with it. So you can kind of cycle through and find any of these, and it'll kind of lead you down kind of a rabbit hole of exploration. So I think it's pretty cool. Sharing there, share back to um, 1 second. Okay. Share. My sharing isn't working now. Give me 1 second. Share. Okay. Okay. There we go. Back. Okay. So, where was I? Key points to emphasize from RoboCop. There's a translate the biomedical questions into graph-shaped queries. It returns multi-hop ranked answers backed by curated sources, and users can explore evidence directly, such as the LinkedIn and PubMed articles. And like I said, it's useful for hypothesis generation and decision support. So what makes a knowledge graph interpretable? Interpretability ensures that different data sets can plug into each other, allowing consistent querying and reasoning across sources. So far, we've seen that knowledge graphs are what they are and how they can help answer complex biomedical questions. But behind the scenes, what makes knowledge graph truly powerful and shareable, and it is interpretability. And it means that the different systems, the databases can work together seamlessly, using shared vocabularies and identifiers so everyone's talking about the same concepts, defining relationships and standard predicates so tools know how to interpret edges, following the FAIR principles, findable, accessible, interpretable, reusable, um, to ensure that data is not only machine readable, but usable across institutions and domains. Interpretability is what lets us connect a disease term from a electronic health record to a gene in a research database, or a drug and drug bank to a phenotype in a model organism study, all within one system. Let's say the system uses asthma, another uses Mondo 0004979, and a third uses C0004096. If these aren't mapped together via shared identifiers and ontologies, then no graph traversal will connect them. So it's important to have that in place so that these kind of things can be shared and be useful. So now that we've defined what interpretability means in a knowledge graph, let's talk about why it matters. First, it enables scalable integration. You can combine clinical, genomic, environmental, and literature data without needing to re-engineer every connection manually. Second, it enhances reproducibility. When you use standardized terms and relationships, others can rerun your queries and validate your results. Third, it supports collaboration. Interpretable knowledge graphs allow researchers, tools, and institutions, excuse me, to work on shared infrastructure, whether they're at the NIH, in academia, or in industry. And finally, it promotes data reuse. The same graph can serve clinical decision making, basic science, um, and policy needs with different queries in the same structure. Imagine if every group created their own vocabulary for disease. One uses asthma, one uses reactive airway disease, and a third uses numeric code. Without interpretability, you never know where anyone was talking about. The road ahead for biomedical knowledge graphs. So knowledge graphs have been unproven their value connecting biomedical data, but the field is still evolving. One big direction is scale. We're seeing more integration of national and international data sets, including patient records, environmental data, and real-time sources like wearable devices. Another is machine learning integration, using graphs not just to store data, but to enable reasoning, prediction, and explainable AI. There's a growing interest in clinical applications supporting diagnosis, identifying drug repurposing opportunities, and resurfacing and servicing risk hidden in siloed data. And finally, we'll need to address challenges like provenance, uncertainty, and governance. Who creates the data, and how do we manage conflicts, and how do we ensure ethical use of the data? The NIH's Bridge to AI initiative is laying some groundwork for knowledge graphs and explainability. Graph neural networks are being built on top of knowledge graphs and more used in precision public health. Integrating social, environmental, and biomedical data is happening currently. From data to discovery, we've seen how knowledge graphs work and what they do. Most importantly, they make data actionable and reusable is what we want to take away. So here are some of the keys that I want to leave you with. Knowledge graphs help us connect the dots across complex biomedical data domains. They are more than storage, they're tools for insight. You don't need to be a computer scientist to use them. With tools like Monarch, Translator, or Robocop, anyone can explore disease, gene, drug connections, generate hypotheses, and surface hidden patterns. Interpretability is what makes this possible at scale. By using shared vocabularies, identifiers, and semantic models, we enable collaboration, reuse, and reproducibility. And finally, knowledge graphs are getting started. As they integrate with AI, support clinical care, and expand globally, they'll become more foundational to the future of biomedical research, I believe. And that is it for me. I will open the floor for any questions and discussions. Um, yeah, thank you.
Now, I will request that Chris opens mics at this time, and you can raise your virtual hands and ask questions. And I must say that I don't see the question and answer box after the recent update of the Teams.
I see it.
I see the box. I don't see the questions. Can you see the questions?
Let's see. I see one question. Yeah, I see the questions. Yeah.
Okay, so maybe you can start from answering the first question in Q&A.
Yeah, so the first question from Stavros, can we get a list of these tools and websites by email? I will provide a, if with this presentation, it'll have all these tools listed at the end of it. So when there's in post-production, when this is passed around, I'm sure that will be available. I don't know who has like access to everyone here. I can drop it in the chat if that would be useful for anyone.
Stavros, so just to clarify, the talk will be posted, the slides will be posted, and it's very easy to find the website. You just type in Google ELK and NIHS, it will bring you to the website, and there is a page for the past events with all the materials from the past. If you have any trouble finding it, you know how to reach me.
Right, and then at the bottom of every slide that had the start of the demo, it had the link to the tool.
Right. So, any questions from the audience at the moment? No, I can start with some. So there is just making sure, I guess, that I understand correct. When you are posting a query to knowledge graph, there doesn't have to be a direct connection between the immediate direct connection between two terms. Let's say you're searching for Alzheimer and the medication, but the medication doesn't need to have an indication for Alzheimer, but it can mention maybe a process that we know is involved in Alzheimer pathology, and there will be a connection still, right?
If there's
depending
no
on how
immediate
it's constructed.
Yeah, so like for example, in the RoboCop example, you can build out layers. So what I just showed an example is I built a single multi-hop or a single hop query. So I went from one node to another node. You can make several of these nodes and combine it. So you can make it as complex or as not complex as you like. If you have a disease or you have a drug and you want to be see if it's associated with Alzheimer's, if that doesn't exist at all, it will probably have a hard time building something for you. But if it's tangentially connected, or if there is anything that's somewhat relatable, they should give you results that say, hey, this is related to these other diseases or other phenotypes that are related to that disease.
Mm-hmm. But in this case, and I don't think you mentioned ranking of the results anywhere. So the distance between that defines the connection will define the ranking of your results, right? The closer the connection, better the ranking. So
so I didn't go into super detail about the structure of some knowledge graphs. So some knowledge graphs you may have seen in the past, they'll have your nodes be different sizes and there'll be a distance between your nodes and the edges that are linked them. Depending on the knowledge graph, the length of the edge can be a type of relation. So it'll say if it's very close, they're saying this has a higher like p-value or something to say that this is more related to something else. I didn't have one of those examples in this presentation, but those do exist. Usually that happens when you are building your own personal knowledge graph using these kind of as a backdrop, using these tools to reinforce something you're currently doing, working on your own research. So a lot of these tools, they have code in the background that you can build onto. They're mostly built on like Neo4j structure. So in my past, I've worked on building knowledge graphs for synthetic population data. And I built that using Python, and then they were ingested into the um, environment of ArcGIS. So doing that is just, it was a certain structure you would provided entities, and then the properties of entities, and then the relationships between those things. Usually, most knowledge graphs that I work with on the day-to-day, the edges aren't too aren't utilized as much as far as using their distances. Sometimes you'll see that more in like word graphs or dot graphs, where they'll take a idea and they'll list other related ideas or terms. Yeah, so that's what exists.
Anything in the Q&A?
Doesn't
Yeah.
seem to be.
I don't think so. Yeah. So I have another question, and that would relate to versioning. So you said that the graph can be extended and it can happen, constantly, right? So you can do the same search today and next week, and maybe receive slightly different results because the data there were nodes added. Right.
Right.
So how is there an appropriate way to understand why the reason for the difference and to document?
Yeah. So having worked with some of the people who worked on RoboCop, I know that the results, as you may get more results, I don't think you'll get less results. I don't know currently if they're in the position of removing a lot of data, unless there's like um a recall on a paper or something like that. So you'll get added results if you did a query now versus a query later. If there's a stark difference between two um, queries, getting the results back. I don't know if there is a mechanism outside of contacting the team that is producing the tool, like Monarch or I saw it. I'll get to that in a second. Like, so reaching out to Monarch or NCATS or RoboCop. They all have ways of going through their own tutorials and ways of help adding to their stuff. So let's see. There's NCATS has it. Monarch has it. Yeah, so for example, I want to say they all have a way to add to, yeah, how to and help to add to their initiatives if your data comes up and, like, say you did a whole separate thing that wasn't in the knowledge graph and you found some information, and you could contact them to add to the knowledge graph. But also, if there is a separation between what you're seeing and what someone else saw, it shouldn't be anything where it's like you're missing the new query should be missing data, but should be adding on the data. Let's see what those questions are. Did I answer your question?
Yes, thank you.
Okay. Okay. Let's see. Okay. Can you implement a knowledge graph on a relational database? Yeah, so um, you can build a knowledge graph using a relational database. However, what you'll be missing usually in a relational database is the actual terms for how they're related. If that exists, then yeah, you can build a knowledge graph pretty much with any um, database or CSV file. So if you have a CSV that has relational database, that is a relational database, you can go on to I'll get the site for you, Neo4j. They have kind of a walkthrough on how to build one. And it's pretty going to be pretty small. I'm gonna pull up Neo4j link and I'll drop it into the chat for you guys. Neo4j. I'll actually share that screen for you actually, so you can see. So Neo4j has a system where you can go in and you can import data. Um, you click a new data source, and they have different ways. So you can have a already have a SQL database, or you have a CSV relational database. You can upload it this way, and this will create an instance that will build a knowledge graph for you that you can then explore. I don't have one ready to go for that particular case, but I'll drop this. And this is neo4j.com, but I'll put it in the chat regardless. Let's see here. There are many ways to build a knowledge graph. Neo4j is the, I guess, the standard currently. But yeah, there are several different tools. If you look up how to build a knowledge graph, there are many tools out there that are setups for building it. There are different tools and different programming languages are. A lot of the tools that I mentioned today have, at least one has a R package that you can download into R. And then Jenny Jenny asks that you highlighted the purposes of translated graph. One function listed on the website is to integrate as many data sets as possible. Do you know if there is a way to filter based on the source, either in the translator system or in the design of knowledge graph more generally? Let's look at it again. So you're asking, is there a way to filter based on source? So source as in like the publications or
Not, can you hear me?
yes.
Yeah, okay, awesome. I guess I wasn't going quite to that level, but more like if you're talking about integrating data sets from different sources, like I don't know if not necessarily an ontology, but like I was talking to Jennifer Fostel earlier today. So like the DTT data dictionary. So if you took information from that source, like if you treat the DTT data dictionaries as an input source, could you then, and that got incorporated into the knowledge graph, is there a way to design the knowledge graph so that, I mean, I sort of already thought of a way I would do it, but I'm not actually, I don't work directly with knowledge graphs. It seems like it would be possible, but so I'm just curious, you have a lot more experience with knowledge graphs. And so my question is sort of like, is that something that can be done in Translator if the inputs have been incorporated from different spaces where it's clinical versus like pharmacy? I'm not sure all of the input sources, but yes, I'm just sort of curious to know if there's a way to filter by source.
Got you. So it depends on the tool, the knowledge graph tool. Some have the ability to filter by the source, others don't. It's just gonna vary from
Okay,
tool to
this is
tool.
a design. Sounds like a design.
Yeah, it
thing.
is.
Okay.
It is. Yeah. Now, if you're creating your own knowledge graph, that is definitely something you could have incorporated. As far as not necessarily the ability to filter, but having it where it you can list out every single source and break it down to where a certain source may be more useful than other sources. You can add weights to things and if that makes sense.
Mm-hmm. Yeah. Yeah. Thank you.
Mhm.
I don't see any more questions. We will give our audience maybe half a minute to come with more questions, and if not, we will thank Taras for his talk and get together for another webinar in a couple of months. And I will just repeat that you can preview previous. You can review previous talks at the website, which I put links in the chat. And you can reach out to us using the emails and on the web pages that I just listed. I think at this, I will say happy Friday and have a great weekend, everyone. And thank you very much, Darius, for agreeing to talk. And have a good sound. Oh, we have a question from Philip.
Oh.
Sorry. Go ahead, Philip.
Sorry about that. Actually, I had a question in there. I'm having trouble with the texting. So you answered my question, Darius, but thank you very much for a very good presentation. Thank you.
Welcome.
Okay.
And I wanted to leave you guys with one more example. What was the video that wasn't in the link that you'll see later, but I don't know when this presentation will go up. So I'm just gonna drop that in the chat for you. 1 second. So I thought it was a pretty interesting video. See if I can get it. There we go. And that's a short video. I thought it was very interesting of a doctor and researcher who had an experience with using Monarch to help diagnose his son.
Yeah. And again, just to remind everyone, last month we had a presentation from Monarch, Harry Caulfield, the Monarch Initiative researcher, and his talk is posted at the website. Well, thank you, Darius. Thank you, everyone. Happy Friday. Happy weekend.
Happy Friday, everyone.